Okay, so Lampbrush chromosomes. So in giant chromosomes, we have two categories. One is Lampbrush chromosomes, and the second one is polytene chromosomes. So let's first talk about the structure of Lampbrush chromosomes, and then we will talk about why the structure is like that and where it is found. So basically, in diploidine stage of prophase 1, okay, of meiosis, what do we observe? We observe that chromosomes, that is your bivalent homologous chromosomes, are attached at a point called as chiasmata. Right, so they are attached as chiasmata, which is like an X-like structure. Now here, what will you observe is that these are your regular chromosomes. Now these chromosomes are going to have points. So say that I make a larger diagram. Yeah, cool. So this is your chiasmata. Now these are going to have points called as chromomeres. Okay, so they have like large number of chromomeres, each and every bivalent. All of them have chromomeres. So they are just, chromomeres are random points. So chromomeres are found in these bivalents, and from these chromomeres emerge out loops. These loops are basically extensions of the chromosome, nothing else. So something like that. So the main axis here of the chromosome is DNA, and on this DNA you have these loops arising like this. Fine. Now if you look at a better diagram, this is what you get. Okay, something like this. So this is basically your DNA filament, just darkening it. Yeah. So this is your filament of DNA. That's just the extension again. Now carefully look. Now this is going to be surrounded by something called as loop hairs. Okay, so something like this. Loop hairs. Now these loop hairs, so this is what you see. This thing you see, these are your loop hairs. What are these loop hairs? These loop hairs are basically nascent RNA molecules. Nascent RNA molecules. Nascent in the sense like newly born RNA molecules. Okay. Now these are nascent RNA molecules, basically your loop hairs. And loop hairs, you know, both the sides, they are surrounded by proteins. Like this. And the inside as well. So they are surrounded by proteins. Now these nascent RNA molecules I spoke about, they are usually mRNAs. They are usually mRNAs. They could be rRNAs and tRNAs as well, but usually they are mRNAs. So these mRNAs and proteins, both of them together, are known as informosomes. Okay, informosomes. Now I said they are found in the diploidine stage of prophase 1 of meiosis. So why are they found? So because what's the point of having so many extensions? What's the point of having so many loops? Because these loops are involved in transcription. So basically it's DNA. These loops, these extensions are basically DNA. Now when this DNA undergoes transcription, we know what happens. RNA is synthesized. Okay, this is through transcription. And when RNA undergoes translation again, what happens? Proteins are synthesized. Now, these cells are highly active, and if they're very active, they need a lot of energy and they need a lot of protein synthesis. And that's the reason, the main reason for having so many loops and having these giant Lampbrush chromosomes is for increasing their protein synthesis. And obviously these loops are, as I told you, they're involved in transcription, which ultimately leads to the formation of protein. So there's high protein synthesis. So that's the main reason. That's the main reason why you have loops. And that's because for high protein synthesis. And these infomosomes I spoke about here, they are used for further development of the embryo. If you're talking about an embryo, which obviously again needs a lot of energy, so Lampbrush chromosomes are of lot of use to it. Okay, it basically it gets its energy because of high protein synthesis. So where are they found? They are found in the oocytes of mammals, vertebrates, and invertebrates. Okay, I wrote something. Invertebrates. Fine. So what are oocytes? Oocytes are basically the mother cells in a cell in the ovary which undergoes meiotic division to form the ovum.